There are two types of immunity, so-called non-specific immunity and specific immunity. And I'm going to talk about non-specific immunity first. As the name implies, non-specific immunity is not specific to particular pathogens. So the white blood cells called phagocytes basically eat up all types of pathogens that have entered the body. They do not really differentiate between the different types of pathogens. They see a pathogen, a cell which does not belong to the body, and they simply eat it up. So what they do is they ingest, and ingest simply means they take it up into the cell, they eat it, and they digest it. And they can do that both in the blood, because the phagocytes float around in the blood, and the phagocytes can also leave the blood vessel and squeeze out into the tissue and attack and eat the pathogens over there. And as I already mentioned, they do not differentiate between pathogens, they attack all of them. And they do that by a process called engulfing, and I'm going to show you now over here a drawing on how this works. Essentially, it's a form of endocytosis. You have over here your phagocyte, and I'm just, it's a cell membrane. I'm going to draw over here the nucleus, and of course the phagocyte moves around. And what happens is, if it encounters a pathogen, let's say a bacterium or whatever, let's say that this is my pathogen over here. What it does is the following: the membrane starts flowing around the pathogen. So we still have our nucleus over here, and you can see that it starts to close on the other side. So I'm going to draw arrows so the membranes continue to close around the pathogen in here, and ultimately we will have something that looks like this. Okay, it's maybe drawn a little bit too big, and the nucleus is over here. You can already see that the membranes are almost touching each other. The pathogen is inside here, and then ultimately we will have over here the pathogen located in a vacuole. That's the newly formed vacuole with a pathogen inside. You might want to label this as pathogen, of course. Always label your drawings, otherwise it's supposed to be an A. Okay, and now what happens next? Now it starts to digest the pathogen inside here. How does it do that? There are small vesicles, and they carry digestive enzymes. And what they do is they fuse together with a membrane over here, and they release the digestive chemicals into the digestive enzymes into this vacuole with a pathogen, and then as a consequence the pathogen, I'm going to just cross it out to indicate that it's digested, and it's gone, and that's basically it. That's the process of engulfing, and you can see it's a process of endocytosis. And just to explain a little bit, because I did not do that first, how can they leave the blood vessel? So I'm just gonna backtrack a little bit and explain to you how they do that. I mean, it's kind of clear over here. If the pathogen is in the blood and the phagocyte is in the blood, then they're gonna meet each other. But how can a phagocyte leave the blood vessel? And what I'm gonna do is over here is I'm gonna draw, let's say, a capillary, and these are the capillary cells seen from the side. And if you have already watched the video on the blood system, then you already know that the capillaries are only one cell layer thick. And that's my blood vessel, my capillary. There are the red blood cells floating around in here. They're pretty large compared to the diameter of the blood capillary. And they're the phagocytes. And what they do is they stick on the wall of the capillary, and they crawl along the wall of the capillary, and you see that between the cells over here, they are also able to squeeze out, and now you have the phagocyte out here. they're also able to squeeze out between the cells, and they are able to attack and do this whole process over here to the phagocyte out here. So if you have a phagocyte over here, it's going to start engulfing it, flowing around, okay? And then it's inside the cell. Okay, so that's basically simply a short explanation of how this actually happens. So we now move on from the non-specific immunity to the specific immunity, and that's on the next foil. And the specific immunity is done by white blood cells called lymphocytes. And what they do is they attack the pathogens by producing so-called antibodies. And the word antibody should have been colored yellow because that's an important term. So that's really important. And the lymphocytes, unlike the phagocytes, are very specific to the pathogen. So that one type of lymphocyte is not able to attack different pathogens, but it's specialized to attack only one type of pathogen. And I'm going to explain over here using some drawings how this works. So first of all, the human immune system starts to make many different lymphocytes randomly. So it just tries out. So we have over here lymphocyte 1, lymphocyte 2, and lymphocyte 3. And how are they different? They're different because on the surface, they have surface structures which are able to bind against different pathogens. And this one I just make it like this. They're much smaller, of course, but for the sake of understanding, this one over here is round, and this one over here I'm gonna make appear like this. Okay, so you see that these are so-called surface antibodies, and what happens here is that they're only able to bind to specific type of pathogens. Now over here I have a pathogen, bacterium, which is actually drawn too big because they're much smaller than the lymphocytes over here, but for the sake of understanding, I hope you get the point. So, and it has a surface antigen structure. Do not confuse antibodies and antigens. It has these things sticking out. Again, they're drawn way too big. Okay. And what happens is the following, that this pathogen over here now enters the body. So that's my pathogen. And enters the body and is now able to bind against one of the lymphocytes that the body has made. So I should draw over here, that's supposed to be the body. And over here, this pathogen now, let's say over a cut in your finger, enters the body, and now the lymphocytes are able to bind against it. And now just look at the structure. Which one of the three over here, A, B, or C, which one is able to bind against the pathogen? Well, it's only B, because only these two structures over here match. So I'm going to draw this over here. I'm going to draw, in this case, pathogen, not pathogen, lymphocyte B. Now, I just realized that's a bad way of actually labeling it, because there are so-called B lymphocytes, and by giving it the name B, you might think that these are B lymphocytes. So I'm just going to number them. Otherwise, because the B has a special meaning, okay? I'm just going to call this a lymphocyte 1, lymphocyte 2, and lymphocyte 3, because otherwise you get confused with a B, because there are B lymphocytes as well. So, and this one over here, as we remember, has this round surface structure over here, and it binds against the pathogen. That's the pathogen. And that's the lymphocyte. And when this happens, it stimulates this particular lymphocyte to divide. So the others don't divide. They're just floating around in the blood at low concentrations. But number 2 over here is going to start dividing now because it was activated. And I'm going to simply indicate this over here, that we now have lymphocyte number 2, and we have now, of course, cell division and then by mitosis, and we have, I'm just gonna draw this very generally, many identical lymphocytes. So that's mitosis. Mitosis is cell division. So only this one starts to divide because this one was activated. The others basically are ignored. So, and now these lymphocytes which divided, they are now able to form, to become plasma cells, and these plasma cells produce antibodies. So we have now over here all of these lymphocytes, and they start producing massive amounts of antibodies, and these antibodies are soluble. They have this Y shape. Again, I am drawing this way too big over here because they're much smaller than the individual cells. And if you look at the surface at the tip over here, you're going to see that they're all round. Now, why is that? Well, of course, because they should be able to bind against the pathogens who have the round surface antigen. So the many antibodies produced, and these cells are now called plasma cells. So what's the difference between an activated lymphocyte and a plasma cell? Well, they become plasma cells. So plasma cells are specific lymphocytes. Essentially, they would just call them plasma cells to make it clear that they produce large amounts of these antibodies. So, and that was it, basically. Very briefly, maybe to summarize, antibodies are these structures down here, the proteins, which are formed by the immune system to fight a pathogen. But the pathogen, and that's something I did not label, has these surface structures against which the antibodies are able to bind, and they are called antigens. And maybe I should complete the drawing over here. This is what do they do, because I did not explain that. You've got these antibodies right now, and how are they able to attack the pathogens right now? Well, these antibodies are soluble. They're floating around in the blood. And now I do not have enough space to draw it, so I'm just going to draw it up here maybe, okay? I hope there's enough space. So you have over here the pathogen, and you have this surface antigen. And the antibody is now able to bind against the pathogen over here, because this fits now. And over here as well, it's able to bind another one over here maybe. Yeah. And if this happens, then this kills off the pathogen. How the killing off works is, again, a different chapter, not relevant here right now. But the binding of these antibodies against the antigens of the pathogen kills off the pathogen. And now you've got these dead cells floating around, and then of course the phagocytes take over and they kind of clean up everything again, because you don't want to have dead pathogens floating around in your blood. So the body's immune system also eats these up. So, and that was basically it. Just to go back to the previous foil, we started off with a non-specific immunity, where you have phagocytes basically eating up everything which is body foreign. And then over here, we have a very specific process in which specific lymphocytes are able to make only specific antibodies to attack specific pathogens. And that is, these are basically the two mechanisms. Maybe I'm just going to add a last thing to clarify this. Forgive me for jumping around. I'm just going to show you how it does not work. Wrong. What is wrong is if you have one lymphocyte over here, okay, making many different antibodies. So this one over here is round at the tip, able to bind against round antigens. At the same time, it makes one that looks like this, like a triangle shape, and at the same time, it makes one that looks has these squares over here, is able to again, and this is wrong. Up, see, I wanted to cross this out. It doesn't work. Okay, now it works. So that's wrong. Okay, so please, that is definitely wrong. One type of lymphocyte is only able to make one specific type of antibody, and that's basically what you have to know. Okay, I was jumping around a little bit explaining this, but I hope you got the point.